And the field of stem cell biology, although it had been speculated for years, needed to have it become more of a science than a speculation about what might be happening. So in 1988, we were able to isolate the first tissue stem cell, the one that forms blood. And I'm going to talk about what's the properties of those cells and where that discovery has taken us over the last 30 years, unfortunately. So, a stem cell is like all other cells in your body, except when it divides to give rise to two daughter cells, one is still a stem cell. So, it is extremely similar to itself. And it's extremely similar because out of the 25,000 or so genes, the daughter cell and the parent cell express the same genes and don't express the others. That property is called self-renewal. So, as you know, there's two daughter cells in every cell division. If one, on average, is still a stem cell, you maintain the number of stem cells in a tissue for life. Following the bombing in Hiroshima and Nagasaki, the people who died at the lowest dose of radiation that kills died, oh, look at that. Wrong, but died because they lost these blood cells. They couldn't make platelets from this big cell, so they couldn't clot their blood. They couldn't make granulocytes or monocytes, so they couldn't fight off infection. It was found in the 1950s, if you put a lead shield in front of a mouse instead of a human and give the same dose of irradiation that would kill off the cells, you didn't lose the blood-forming system. That led people to know that the bone marrow was the site of blood formation. There were a lot of speculations about how you make blood. In fact, many people thought, that must be an automatic, that you must have one stem cell to make this cell, another one to make red cells, another one to make B cells, another one to make T cells. So we needed to purify the cells so we could act as if we were biochemists and determine the properties of those cells, and then transplant the cells, like in a bone marrow transplant, to see how they function. So, what we found, there was a single time of development of blood-forming stem cells. In biologist terms, it's when the embryo to fetus transition occurs, which means when you go from having cell types that will not stay in your body, but they must be formed to make patterns of tissues, to actually making lungs and blood and brain and bone and so on. Stem cells are only born at that stage. Blood-forming stem cells only born once. So for the rest of your life, you've got to have those cells both to maintain the system and when things go wrong, like leukemia or a bad blood disease, like what's called, I'll get to it, myelodysplastic syndrome. You should look first to see if it's a problem in a stem cell and how it could be a problem, even starting with a single blood-forming stem cell. When we started this, we were examining the blood-forming tissue like bone marrow or what's called mobilized peripheral blood. Don't do that, you're giving away the whole thing. Ooh. Yeah. So, in the bone marrow and the mobilized blood, you have a mixture of 1 in 100,000 cells being blood-forming stem cells, and all the rest being forming blood. But if you have a cancer that's spread through your body, like metastatic breast cancer, most of the time we found It was also in the bone marrow and the blood. Many, many years ago, like in the 90s, people tried to cure patients with metastatic breast cancer or hard-to-treat lymphoma or myeloma by giving them more than a lethal dose of a combination of chemicals and/or radiation. So if chemical A kills 90% of the cells at 1 milligram per kilogram, at 2 milligrams per kilogram, it'll probably kill 99%, 3, 99.9, and so on. But when you know you have leukemia, or when you know you have metastatic breast cancer, you have at least 100 billion cancer cells in your body. And if it's spread, for example, beyond the breast to the blood, like I'm mentioning, you can't use surgery anymore because it's not localized. You can't use radiotherapy anymore because it's not localized. So you're stuck with trying to increase the amount of chemicals you give that are very toxic and have very bad side effects, but which kill more and more cancer cells the more drug you give. The limitation was you ran out of blood formation. So in order to get around that limitation, the bone marrow or the mobilized blood was taken out and frozen down. Then the patient was treated with very high doses of combination chemotherapy, and then this was thawed and injected back in the body. But as we found out, most of the time, women with metastatic breast cancer, that's at least 60% of the time, have cancer cells there. So if you reinfuse cancer cells into a body that had been depleted of its blood-forming and immune system by chemo or radiation, you were giving cancer cells back to the body. And as we showed later, people died of the cancer that was injected back in. So we wanted to find a way to get around it. And so we developed this way of purifying blood-forming stem cells, thanks to the Herzenbergs and the development of a machine that can sort cells based on their properties. I'm not going to go into the machine, just to say it's a very neat machine. And we could sort the cells, and since we had molecules called antibodies that could bind, let's say, a red-colored antibody to half the cells in the bone marrow, we could sort into red and not red. Then we could do it again with blue and not blue, and green and not green, and so on. And finally, we found this 1 in 100,000 cells that only contain blood-forming cells. No cancer cells, no T cells. And this we tested directly, 250,000-fold depletion. So I helped form a company called Systemics, and at the company, beginning in the 1990s, we treated women with metastatic breast cancer here at Stanford. And there were two groups of women who were treated. They came in with the same criteria. They got the same very, very, very high dose of triple combination of drugs that would kill them. But before they got that combination, we isolated and purified either their stem cells or those others that got the standard of care got back the whole mobilized blood, which we know in 60% of the cases had the cancer cells in it. At about this part of the curve that I'm going to show you, and I'll explain in a sec, a very large company purchased my company saying, we know that you don't have enough money to do all the clinical trials you want or need to do. Whether it's regeneration of the blood-forming system after you kill it off with high-dose chemotherapy, or you have people who are radiated by accident and need to have a blood-forming system, or you have a defective blood-forming system and you want a healthy one back, or you don't want to give all the drugs to immunosuppress a liver graft or a heart graft or a lung graft. All of these I'm going to show you we could approach if we could get pure blood-forming stem cells. So here's the results. Even though they bought the company at that time point, shut it down at about that time point, we could retrieve the data from the trial. So this is the overall survival versus the time after transplant. And what you can see in this curve is that half of the women, 50%, were dead in about 26 months if they got the standard of care. That's the level then, and that's the level now. No improvement with all the kinds of drugs or hype or hope that people have. But as we went back and looked at all the data, we didn't reach half of the women dying until 10 years. And now, actually much more than this, 21 to 23 years after the therapy, 1/3 are alive without disease. I interviewed 2 of them this year. And the one thing they told me, only when I interviewed them, was their Stanford breast cancer doctor begged them not to go into the trial. So I want you to think about that, because that's an issue. It's maybe the biggest issue I can get across. So this therapy is not available. Although about 5 years ago I negotiated with that very large company and got back all the methods and the ways to start over again. Not only for these ways of working with stem cells, but others. And put it in Stanford Hospital and Medicine in a not-for-profit setting. And we've applied for funds to make the reagents for any bone marrow transplant group in the world to be able to separate stem cells at cost or for free, depending on who gives us the grant. So we're going to try to re-energize this field because that large company, which shut it down, which promised they wanted to do everything we did, found a compound you could take by mouth that would treat one disease and make $2 billion a year. It wasn't that they were against us, but they had bought up lots of intellectual property and things that were going to clinical trials, and they had a responsibility to their shareholders to make the most money they can as fast as they can. Don't think that that's different. They're not a philanthropy, they're not the National Institutes of Health. Any company that doesn't do this, took me a long time to learn this, but any company that doesn't do it won't be a company after the shareholder suits that they aren't maximizing the profit of their investment. So we have misplaced our trust in large companies, large pharmaceutical companies, and we have not taken on the responsibility within academia to carry things all the way through early phase clinical trials, like this one, and keep it at your academic institution so that you can then license it out, and you know you don't have risk, because they don't want to take risk, and you have some evidence of efficacy. I'm taking a little longer, I'll move faster. So we wondered whether in all of the blood-forming system we could solve how a cancer of the blood occurs, how it happens. So we took patients with acute myelogenous leukemia, and we began to look at not only how the leukemia develops, but Ravi Majeti and I and a whole group of others found every step from the blood-forming stem cell to blood. And we could isolate every one of those cells. So now we could ask the question, if there are mutations in a particular patient's leukemia that are involved in the development of the leukemia, is it in the blood-forming stem cells or one of their daughter cells. And way back in the year 2000, two Japanese scientists and I got bone marrow from Hiroshima Hospital of people who got leukemia 10, 12, 15 years after the bomb. And we found that the leukemia itself, that's what this is supposed to look like, which had undergone somehow all of these changes, was at the level of a multipotent progenitor, not a stem cell. Now normally, it's only the stem cells that can self-renew. Multipotent progenitors, the next way down, they can make blood, but they can't self-renew. So they only do it for about 6 to 8 weeks. But the leukemia, all of the leukemias, luckily for us, were at that stage of development. So this is a theoretical set of the kinds of mutations that people have been uncovering in leukemia patients. So if there were, for example, 7 of these events in the leukemia itself, we proposed that there had to be a cell that had only 6 of the 7, or 5 or 4 or 3 or 2 or 1. That's what we wanted to understand. And when Ravi Majeti and I and Steve Quake, a physicist who's here now at the Clark Center, got together, we sequenced 21 straight leukemia patients to find the mutations. And then we made a DNA test for each of the mutations. Patient number 70 had 21 mutations. And then we took one blood-forming stem cell at a time from their same bone marrow and asked, did it have 0 mutations, or 1, or 1 plus 2, or 1 plus 2 plus 3. So as scientists, we could actually start to ask, how did the leukemia happen? And if there are mutations, do they have a particular order that they occur? Which would be clues to tell us what events might be important to develop leukemia. This patient actually had 21 mutations. And the very first mutation, which had occurred originally in only 1 cell, had grown to maybe 10 or 20,000 cells, and had spread throughout the body, so we could stick a needle in any bone and find that cell with that single mutation. Now in our body we have, for all of the genes we express, we have some from mom and some from dad. So if that's from mom, the very next mutation was the same gene from dad. And both of them lost the function of the gene. And just to show you it wasn't an accident, the very next patient we looked at, same thing. But they didn't all follow exactly the same pattern to get to leukemia. The other thing is, as those cells expand, they expand at the expense of normal stem cells. And that's because we showed several years ago, blood-forming stem cells have a home in the bone marrow, a structure that holds probably just 1 stem cell. So in order for the pre-leukemic stem cells to start expanding, they competed with the normal ones. So that's an important point. And at the very end, the final mutation for the aficionados, FLT3 internal tendon repeat, or KRAS, or NRAS, or beta-catenin activation. Always the last one, and it causes proliferation. So this is the setup to the leukemia, and then this is the emergence of the leukemia. Every leukemia, every myelodysplastic syndrome, every myeloproliferative neoplasm. I'm just using the word Stocks used. If there's anybody in the audience who's a doc, that's what happens. And not many people know all this. Here we go. So we showed that in acute myelogenous leukemia, all of the progression up to the leukemia is in the blood-forming stem cell. But the leukemia emerges at the next 1, 2, or 3 steps down. So it doesn't require the niche, the home. It's driving on its own, and it has essentially unregulated cell division till it kills you. There are no leukemias that we found ever in mice or humans that are leukemias of the stem cells. So that home regulates the number of cells that can be in that home and have that stage of development. Really important. We don't know how yet it works, but we plan to find out. Now, it turns out, as we and others have shown, every organ in our body, looked at closely, has its own organ or tissue specific stem cell. My wife and I in Nabucco Uchida in 1999 discovered the human brain stem cell. And we get isolated, and I won't go through it, but we went through lots of clinical trials for spinal cord injury, congenital genetic diseases, and dry age-related macular degeneration. So we know, and they make brain and only brain, and the blood-forming stem cells make blood and only blood. So we knew we were onto something. So we know that all those tissues have it. So if the mutations that I showed you here, and there's a lot of different names there, are found in a blood-forming stem cell clone, we actually asked the question, could that mutation or another mutation turn a non-blood stem cell into a self-renewing cell? And none of them could. So that's why they hitchhike in stem cells and form what we call a clone. Daughter cells that have it. And about 2/3 of the mutations are not part of the leukemia, they just happened. And 1/3 are driving the bad behavior of the leukemia stem cell when it finally emerges. So, we think this is how all cancers develop. And it means we have to find, for breast cancer, the normal breast stem cell. We have already the brain cancer, for, the normal brain stem cell. But you have to look at the lung stem cell to see, is there a cell in between the normal stem cell and the cancer cell that shows you the steps in between, and will each of those steps reveal a new vulnerability to treat the cancer? I won't say that for a while. Actually, a minute. So if this is the process that leads to the leukemia, if you fall short of it, you still can get a stem cell, as shown by Wendy Pang, who's now an instructor at Stanford, that takes over all the blood-forming stem cell homes. But when it tries to make the blood cells, they die. They actually are eaten by scavenger macrophages patrolling for rogue cells that might have some abnormality which can be detected and used to get rid of it. Sid Jaiswal, who was actually with us for his graduate school and medical school, and then went to Harvard, everybody makes a mistake every once in a while, showed that if he looked at the bloods of normal people, apparently normal people, as they got older, he would find every once in a while a clone of a cell that had expanded dramatically, but there was no known disease. Except when they then looked in retrospect, they had a higher, let me just go back one point. It takes a long, 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 long time to accumulate 7 mutations. If 100 is the number of people who get leukemia, it's probably a couple hundred or 1,000 that get this disease. And this one, maybe 10,000. Almost certainly there's somebody in this room who has that going on. But presumably you won't make it all the way to leukemia. But part of the fascinating and unexpected things that we found is that those people not only have a higher chance of getting the blood diseases, it is the highest risk factor for heart disease, myocardial infarction, stroke, aneurysm that blows up the aorta. Highest risk factor. So that was what studying the disease in clones of stem cell told us. It doesn't tell us yet how it works. And so Sid and Nick Leeper and others are working on how to follow that up to treat cardiovascular disease, which everybody thought was, and many people say still is, a disease simply caused by eating the wrong foods, not exercising, and so on and so on and so on. Well, that might be true, but there's also a genetic basis unrelated to that. That's a very high risk factor. When we looked carefully at the leukemia stem cell, and we compared it to a normal blood-forming stem cell by a method that asked which genes are expressed at high levels, and the leukemia but not the normal, we found two things. That the leukemia always had a molecule called, it's not my fault it's got this name, CD47. And that turned out to be a don't eat me signal for those scavenger macrophages trying to get rid of rogue cells. So if this macrophage comes upon this cell, it can't eat it because it's paralyzed by this CD47 interacting with this macrophage molecule that is bound by it. We also found later, actually my daughter, when she was in high school, shows you how tough this is, an eat me signal called calreticulin. Doesn't matter what its name is, but it's this balance of don't eat me and eat me that determines whether that will be gone. So we wanted to know, is that CD47 molecule really important? So we took human acute myelogenous leukemia cells out of the bone marrow of patients. We put them into the bone marrow of mice that have no immune system to reject them. So they're like the bubble boy. Have to live in a bubble, no immune system. And we waited. And by 4 weeks, the bone marrow, which isn't supposed to look like this, just has visible leukemia in it. Just like at the worst trace of a human disease. We treated it with a new antibody we made that blocks the interaction between the don't eat me signal and the macrophage receptor we bind to. In 90% of the cases, we cleared it. 10% of the cases, the macrophages, the eating cells, are eating like mad, but they can't catch up with the tumor. So, this actually has moved quite a long way. There's a company that we helped form, which made that antibody in very high amounts. Tested to make sure it wouldn't be toxic. Took it through early clinical trials, and now here's a clinical trial led by Mark Chow and others, where we're looking at the leukemia in patients, or that disease I called you myelodysplastic syndrome. This is the beginning of treatment, and each of these bars is a patient. If the tumor kept growing and go up larger, it goes up. If it regresses, it goes down. So far, at least at this time point, they're all going down. This is a discovery you couldn't have made any other way. You had to have pure blood-forming stem cells, and you had to purify the leukemia stem cell so you knew the relevant targets to go after. That made us think, well, how do the other antibodies that help cure cancer work? So the most famous one, largely helped in its development by Ron Levy here, is called Rituximab. Rituximab binds to a molecule on a B-cell lymphoma called CD20. And it helps eliminate the tumor. We showed directly that when the antibody binds to that molecule on the lymphoma, the back end of the antibody binds to something very tightly that takes the lymphoma cell into the macrophage and kills it. So we said, that's good, but not all the patients are cured with rituximab. So we combined the blocking antibody to the don't eat me signal and added the antibody that helped make a stronger eat me signal. Here's the results of that clinical trial so far. These are patients who had failed all other therapies with either follicular lymphoma or large cell lymphoma. And about half of them have gone into response, and about 20 to 30% overall so far are a complete response. So we think that this lead could be quite important. I know I'm getting close to the end. But 3 or 4 years ago, a young cardiovascular medicine physician named Nick Leeper called me up and he said, you know, I've been looking at samples from people who died of heart attacks. And I've taken out the atherosclerotic plaque, the area that got blocked. And I did a gene expression test on it, and CD47 and calreticulin are there. So it's a long story, I'm not going to go on, except to say that we found that the smooth muscle cells in an artery from a single, probably mutant precursor expand, and they express the don't eat me signal. They cause inflammation, the big scavenger macrophages come in, but they can't eat it. So we did a test first in mouse, where we picked a mouse that on a high-fat diet would die of a heart attack or a stroke or an aneurysm in 2 months. And we treated it just with a net blocking antibody to CD47, and that worked out well also. It also works for fibrotic conditions. If you've ever had surgery or an infection, for example, in the uterus or the fallopian tubes, a secondary effect can be adhesions. And the adhesions can glom up the bowel or the uterus. And again, Jonathan Tsai, now to make a long story short, showed that we can clear an adhesion in a mouse model with the anti-47 antibody plus another antibody. And Gerlinda Vernick has shown in pulmonary fibrosis, which is currently incurable, that she can make a model in mice of the human disease, an anti-47 plus an anti-inflammatory antibody,